Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi taala wabarakatuh. We are from group Critic 2. So I'll start the presentation. Our stand on the case study is research or study on germline gene editing must not be banned, which almost similar with presenting in group Critic 1 stand. However, to, to continue with the study, the condition must be really strict in several aspects such as the ethical approach, method or procedure used throughout the study, and importantly the purpose of the study itself. Because the study on gene editing can really be a great potential for the medical purpose in the future. Okay, then we'll proceed with other arising matter throughout this case study, inshallah.
What CRISPR baby prison sentence mean for research? Chinese court sends strong signal by punishing He Junqiu and two colleagues. This is the highlighted title of the article that told us about the Chinese scientist named He Junqiu, who was sentenced by Chinese local court to 3 years of imprisonment and fine, with his two colleagues for committing the crime of illegal malpractice when conducting gene editing on human embryo to prevent transmission of HIV. He Junqiu action opened the eyes of the researcher and public about the legal and ethics on human gene editing. The experimental procedure conducted was unclear, non-transparent, and may cause irreversible damage to the entire of human genome. After He Junqiu cases, Chinese government promulgated the Regulation of People's Republic of China on the administration of human genetic resources, which aim to protect public health, national securities, and public interest through effective protection and rational use of China's human genetic resources. Not only that, He Junqiu action also brings wider chilling effect to other researchers who use CRISPR as tools of the study. They worry that it might be difficult to get approval to use gene editing tools in clinical trial, including use of tools to edit adult cells, which does not raise the same ethical question as work in embryo. Moreover, some scientists worry the startling claim that lead to knee-jerk regulation that might cause countries still working on regulation that make it unnecessarily hard to do the research involving gene editing. In addition, He Junqiu action would damage the public trust on gene editing. However, the He Junqiu action led to moratorium on gene editing in embryo and germline cell, as it would discuss on safety issue and broad societal consensus about relevancy of gene editing and germline cell. So, some of the researchers feel triggered to conduct the research to edit the genome of embryo when the regulation is ready to abide by.
As for my part, I will explain on the differences between germline genome editing for disease prevention and for enhancement. Germline genome editing for disease prevention involves the correction of genetic defects to improve or cure genetic diseases. Meanwhile, germline genome editing for enhancement is a non-therapeutic intervention intended to improve individuals with desired human traits. So with germline genome editing for disease prevention, the embryo should have a defective gene. As for enhancement, the embryo is normal but then edited to enhance its natural function into something special, in this case was done by He Junqiu. So even though Dr. He said that his purpose in doing germline genome editing was to prevent transmission of HIV, what he really did was genetic enhancement. So is there any implications of genetic enhancement? Yes, there is. With germline editing for enhancement, there are so many complicated, uncertain or even unknown factors that may impact the genetic structure and development of an embryo. And these factors will make it more difficult to identify the risks and benefit of the technology. So without being able to assess the risks and benefits of genome editing, the information will not be available to volunteers, and therefore they will not be able to make decisions about whether to give consent, in which eventually led He Jiankui to forge the consent document. The application of concepts and tools of genetic enhancement may lead humans to design and reproduce more desirable and better kind of human beings, that is also known as eugenics. This will tempt people to create babies according to their preference, which is considered immoral.
Assalamualaikum warahmatullahi wabarakatuh. Now, we move to the potential application of gene editing techniques and its benefits. From the previous group, the presenter had explained on the scientific approach of gene editing techniques. However, our group would like to add some points on the principles of the potential application of this method. There are two possible applications discussed at the moment. The first one is the infertility treatment and the second one is disease prevention. As we know, with the arrival of new methods of genome editing, new perspectives on germline interventions have arisen, including the application of this method for infertility treatment. In germline gene techniques, all elements of the germline such as oocytes and the sperm cells can be edited. For example, after the retrieval of oocyte from the mothers or donors, the method could be used to correct the mutation in the TUBB8 gene, which is known to cause developmental arrest after fertilization. In addition, sperm cells also can be edited through spermatogenous stem cells editing. By conducting a testicular biopsy, the assays can be retrieved and transfected with programmable nucleases and trigger the production of mutation-free sperm cells. Therefore, this procedure have the potential to be used to treat genetic infertility. For disease prevention, germline gene editing could be a therapeutic option for several hereditary monogenetic diseases such as Huntington disease, beta thalassemia, and many more. In an article, it was reported that by using germline gene editing method, the genetic mutation in MYBPC3 gene can be corrected in the zygotes and led to the development of mutation-free embryos. However, in this research, the embryo were not implanted in the utero. Nevertheless, this study shows that correcting a gene mutation in viable human embryos using genome editing method is feasible. All in all, germline gene editing can benefit the population by first, the method have potential in helping infertile couples to reproduce, second, disease prevention in population, and third, to increase longevity and improve quality of life. However, there are still risks and assessment needed to be done thoroughly for the method to be safe to use. Therefore, next my friend will discuss some risks and consequences of germline gene editing techniques.
Next is the limitation or cons of genome editing that may be encountered by the researchers along the work process. The first one is that this genome editing is a challenging experimentation as it needs a very delicate work with high specificity and precision. The researchers who are going to do this experiment must have an adequate qualifications and credibility in order to avoid any mistakes in the process of creating the new genes. Apart from that, this genome editing may also involve the procedures that was forbidden by the law. For example, this case of He Jiankui, who did the experimentation to the embryo for the reproductive process. High specificity and precision of work is needed in handling this genome editing as there is a risk of mutation to happen. For instance, when the enzyme was introduced into the body, it does have the targeted gene, but in our body there are many similar looking DNA region and there is a possibility for the enzyme to go and bind at different gene. And this will lead to the second cause of genome editing, which is the risk of mutation in the edited genome. This may be happen if the enzyme go and cut off the wrong target, thus causing the production of other type of protein, which may lead to another disease or unintended harm. The last limitation of genome editing is about the continuity of edited gene to the future generation. It would be good if the experimentation was successful and the gene carried throughout the generation were in good manner. However, there might be a risk in which the error or manipulation occurred in the edited gene where it will be passed down to the next generation. If any error present in the gene, it will live and blend well in the body environment, and this is known as genetic drive. If there is any mutation happen in the next generation, it will be very difficult to be controlled. Also, the researchers need to keep updated with the future technology involving the genome editing, as the current technology might be different with upcoming technology, and that may also causes the changes in the ways of treating any mutation that happen in the future.
So, how much do we need gene editing? As in his cases, he used germline gene editing, which altered the entire embryo to be less susceptible to HIV, and this technology will affect the future generation. However, this germline gene editing was different with the somatic gene editing, as it only modified a potential DNA to cure or treat the disease by genetic mutation, and somatic gene editing will not affect future generation. As for now, germline gene editing is still new, and there are many ethical issues need to be considered. So the usage of this germline gene editing should be halted for now. While researchers can use somatic gene editing to find a solution on the treatment of the HIV infection, as this technology had been researched for the past 20 years and highly regulated. Besides there are less ethical issues need to be considered by using somatic gene editing.
We move to the next point, which is there any limitation on the usage of advanced biomedical technology in Islam? Generally, Muslim practice advanced biomedical technology is as same as West, but the ethics may be different. Firstly, Islam does not condone the use of advanced biomedical technology as long as it abides with Islamic principles that have been discussed in the previous group before, which is Maqasid Syariah. In regard, the original ruling of advanced biomedical technology is permissible in Islam but can be changed to dislike and impermissible guided by Islamic law. This means Islamic law play a significant role on encouraging the usage of advanced biomedical technology through the fatwa, which always condone the usage of advanced biomedical technology while placing limitations on some practices. Thus, to relate with this technology of CRISPR baby gene editing, Islam limits the technology as it offers less benefit over harm, which is not aligned with Islamic principles.
As for my part, I will explain more about how to educate oneself with adab and ihsan instead of using laws and regulations to educate oneself as a Muslim. So, by definitions, ihsan is a method of taking one's inner faith, which is Iman, and showing it in both deeds and action. And the actions, we call it as adab, is the refinement and the good manners, and also the morality. To incorporate adab and ihsan into ethics, so basically Muslim scholar has been viewed ethics into two parts, which is the first one, ethics as a character, which means the state of soul, and the second one, ethics as science, which means state in which character become object of its inquiry. So here I will be focused more on ethics as a character to be developed. So we have a few things to be discussed, which is the first one, we have principle in Islamic moral system. Second one, we have traditional Islamic approach, and the third one, we have modern Islamic approach. Basically, principle in Islamic moral system, we have 12 principles, but then there is one very crucial here, which is the moral property of given conduct. So in moral property of given conduct, Islam has been underlined three important things, which is the first one, niyah solehah. We must have good intention to help the people. The second one, we have wasa'il mubahah, which means permissible means, or we call it as permissible way to conduct the study. And the third one is amal tayyib, which means the desirable outcome. we want certain outcome, certain positive outcome in the results of the study. Next, traditional versus modern approach of Islamic medical ethics. So as for traditional, as we can see in his case, the first one, qasad, intention. So intention, he has a very good intention, which means he want to protect against the HIV for the baby. But then the second one, yakin or uncertainty. So the safetiness of the CRISPR-Cas9 procedure is very uncertain. And then the third one, darar, which means avoid harm. The risk is still uncertain. And next, for the darurah, which means the necessity. So the necessity of doing CRISPR-Cas9 to the baby is still questionable. Is it necessary or not? And next, uruf, which means the social acceptance of the Chinese public or Chinese netizens is still low, which means the public cannot accept the germline gene editing. So next, for the modern approach of Islamic medical ethics, we have first ihsan, doing good or beneficence, doing good towards others. The second one, we have siddiq and amanah, which means truth and trust, which means we have to have qualifications to do the study. And then the third one, la dara wa la dirara, which means haram may be neither inflicted nor reciprocated, which means the lesser harm will eliminate the greater harm. The next, the fourth one is hifzus sir, which means the confidentiality of the informations for the patients must be not exposed to other parties. Next is adalah and qist, which means justice and fair. So regardless of the qist, regardless of the who we are treating, we must be justice, we must treat them fairly. So the next one is bayan and adamul wish, which means clarity and full disclosure, which means before doing this study, we must disclose the information towards the volunteers or towards the patients regarding the risks and the benefits and the consequences of doing this study. Next, the last one is to respect the patient's autonomy. So as a researcher, we have to respect whatever decision made by the patients.
For the general recommendation, we suggest a full set of laws with specific regulations and guidelines should be formulated to punish the misconduct in genome editing behavior and to prevent similar negative events in the future, as the laws are the last line of defense for social governance. Therefore, further recommendation for further legislation are as followed. Firstly, a specific crime and more severe penalty should be formulated in the Chinese criminal law, as the current Chinese law are inadequate to deal with the new challenges due to the new expertise and technologies. As we are informed that in his cases, he only being punished for 3 years imprisonment, while if we followed the punishment of similar behavior based United Kingdom law could be 10 years of imprisonment. Secondly, is to have a better governance where the local government and other administrative agency should know and understand their responsibilities if they fail to carry out their duties in ethical review, supervision and management of people under their management. Thirdly, more effective and binding mechanism to constrain the use of genome editing technology should be developed, where more specific guidelines and preventive measures should be formulated inconsistent with the international regulations. Lastly, a collaborative network should be strengthened for better global registry and surveillance of human genome editing technologies and research, where the expertise such as the WHO Expert Advisory Committee on Developing Global Standard for Governance should lead this program. To add, the existing national legislation fails to provide the governance framework necessary to regulate germline engineering in the CRISPR era. They have released more than 60 ethics statements on germline engineering, but there is still no international consensus on how human germline engineering should be regulated. For your information, these are the reasons why regulations on germline research are particularly challenging. First, the law struggles with capturing the complexity of the embryos as emerging biological entities, as there are different legal systems with different definition on which point an embryo exists. Such as in the United Kingdom, their legal references to an embryo is including an egg that is in the process of fertilization, or it is undergoing any other process capable of resulting in an embryo. While in Switzerland, the embryo exists after pronuclear fusion has taken place. Thus, some country avoids the confusion by just regulating embryo research without defining what the embryos are. The regulation of research using gametes is relatively underdeveloped. There are very few rules applied specifically to the use of sperm and oocyte in basic research. Third, most regulatory framework neither prohibit nor permit germline genome modification specifically. They regulate research with embryos, but they are silent as to whether researchers can modify gametes and embryos or not.
HIV is a preventable disease, and as the saying goes, prevention is better than cure. So how do we treat the root cause of HIV? The answer is by education, which is the most cost-effective ways in preventing this disease. So there are two aspects of education. First is education for HIV prevention, and this is to prevent new infection from taking place by giving information and knowledge revolving HIV, such as the routes of transmission, blood risk, implementing sex education, and also giving information in preventing infection in the medical settings. The second is education to mitigate the effects of HIV. So in certain parts of the world, such as India, the HIV positive people were being burned to death. And in China, this group of people were facing multiple discrimination and stigmatism from the community. So having a good quality of education helps to mitigate the negative consequences of HIV by addressing the issues of rights for privacy, dignity, and freedom from discrimination. It also allows people, especially the HIV positive people, to understand the rights to have access to good healthcare, education, and social security.
Next, without using advancement technology, we can also look back on the Islamic teaching as one of the solutions to prevent HIV infection. First is to never commit a wrongful sexual act such as adultery, sodomy, and lesbianism, as these acts will lead to the HIV infection. Allah also commanded in Surah Al-Isra verse 32, and not draw near adultery, as for it is a shameful deed and an evil way leading to destruction. Second, Islam also forbids us to abuse drugs, as one of the way for the spread of HIV infection is through the sharing of the needle when you abuse the drug. And lastly, Islam also asks us to always practice a good hygiene, such as washing hand immediately if you came into contact with the blood or liquid from the HIV patient.
Her Janku's actions has somehow made us ponder as a Muslim whether his conduct was merely pure evil and why Allah allows bad things or evil to happen in the first place. So let us understand about what is pure evil. Pure evil is defined as an independent phenomenon whereby no dimension of good is involved. There is nothing in our existence that can be called pure evil, because every evil in this world is good from one angle or another. Allah created them for the greater good, as their existence outweighs the good of their inexistence. For example, without evil, it will become difficult for us to recognize Allah's attributes and qualities, such as a forgiveness and pardon, as well as justice and dominance. Besides, evil also acts as a test for us, which is to test for our conduct as well as to test for our faith. As Allah mentions in the Quran in Surah Al-Jinn verse 35, every soul will taste death, and we will test you with evil and good as a trial, and then to us you will be returned. With these tests, only we will be able to realize our dependence on God. Also, without evil, scholars will be incapable of deliberating, verifying, and learning. Thus, no knowledge would exist. To relate with He Janku's action, his evil conduct has somehow become an eye-opener for the other scientists, ethicists, lawmakers, as well as scholars to revise his actions and put things right, such as by reviewing the existing laws and ethical regulations and bring justice to the victims.
Alright, everyone. So for the last part, to conclude, research or study on germline gene editing must be continued for the sake of knowledge and medical advancement, which have a great potential in curing the disease in the future. Uh, However, this must be followed with a strict protocol uh, to avoid any bad intention, unethical research, and even illegal medical use. That's all. Thank you.